All right. So just a reminder to everybody that our test is this Friday. The material we end in this PowerPoint. So whenever I'm done with that PowerPoint, or this PowerPoint rather, that's the end of the material. As always, go to our test 2 folder on Blackboard to see what is and what isn't on the test. When we move on to our new PowerPoint, that will not be on the test. All right, so on Friday, we ended talking about BPG, how BPG can bind to hemoglobin, and BPG forces hemoglobin to adopt the T state, the 10th state. Just remember, the T state does not want to bind oxygen. The R state does want to bind oxygen. And we talked about all the different things BPG can affect. It can affect how you deliver oxygen at different altitudes. And it also helps affect how fetal hemoglobin can outcompete adult hemoglobin, so the fetus can get oxygen from its mother's blood supply. All right. So I have a question here. We can skip it. It's just a thought experiment. So we can skip question 3 here. So no need to go over that. Question 4 is one I want to get to because it really does test our understanding of how hemoglobin works in its different states. So there is a human globin mutant called Rainier, in which tyrosine 145 on the beta subunit is replaced by a cysteine. When we have this mutation to cysteine, we form a new disulfide bond with another cysteine that's in the same subunit. So we have a mutant disulfide bond. This mutant disulfide bond prevents the formation of ion pairs that normally stabilize the T state. So how does Rainier different from normal hemoglobin with respect to oxygen affinity, Bohr effect, and the Hill constant? All right. So Rainier, before you answer that, so it prevents the formation of ion pairs that stabilize the T state. So Rainier is usually, like always, in the R state. So how does that affect things? And I should have a poll here. Let me see. Let me just edit this poll super quick because I just realized there's a typo in it. So give me 1 sec here. Edit that. Edit that. And edit that. All right, let me launch that poll now. All right, so how does stabilizing the R state, which Rainier does, affect oxygen affinity, the Bohr effect, and the Hill coefficient? So everybody take a little bit time to review what those terms mean in terms of hemoglobin, review what those terms mean for T and R state, and see if we can't come up with answers here. All right, so we have about half of you right now. So, okay. Give about 30 more seconds to think this over. All right. So let's take a look at this now. So how does stabilizing the R state affected, well, I guess there's more than one typo. How does stabilizing the R state affect oxygen affinity? Oxygen affinity increases. That's correct, right? So the R state wants to bind oxygen, right? So if I want to bind oxygen all the time, then my affinity for oxygen will increase. I always want to be bound to oxygen. Basically, in Rainier, I really can't go back to the T state, so I always want to bind oxygen. How does stabilizing the R state affect the Bohr effect? The Bohr effect has less of an effect. That is correct, right? So the Bohr effect, right, really works by transitioning from the R state to the T state, right? So when pH becomes lower, what will happen is that hemoglobin will bind a hydrogen. When it binds that hydrogen, it will go to the T state and it will release that oxygen. Well, if I have a mutation that does not allow me to go to the T state, then pH won't matter. I won't go to the T state. So the Bohr effect is going to be less. Hill coefficient. What does Hill coefficient do? So we had almost an even split between increase and decrease. Hill coefficient will decrease, right? So before I explain that, does anyone know what value the Hill coefficient will go to if we're stuck in the R state? Does anyone know that? Won't go to under 1. Under 1, less than 1 means negative cooperativity. Negative cooperativity means that if I bind something, I don't want to bind it. If I have multiple binding sites, I bind one ligand, my other binding sites don't want to bind the other ligand. That's not what quite is happening here because we're all in the R state, right? Greater than 1 means positive cooperativity. That means if I bind oxygen in one site, my other sites want to bind oxygen. So that happens when there's a T to R transition, but we're always in the R state, right? So it won't ever transition. So it's not over 1. It's not less than 1. It is 1. 1 means no cooperativity. So the correct answer is the Hill coefficient will decrease because in normal hemoglobin, Hill coefficient is like 3, right? So normal hemoglobin has a lot of positive cooperativity. The way that positive cooperativity works in hemoglobin is that if we're all in the T state, right, and one T state binds oxygen, so O2, then that will go in the R state, whoops, and that trigger to the R state can help make the other subunits go to the R state, and now they want to bind more oxygen. So that's how we have positive cooperativity, right? But if I'm already all in the R state, I'm at the maximum amount that I want to bind oxygen. So if oxygen is around and I bind oxygen to one subunit, the affinities of the other subunits are unchanged. I'm already bound to oxygen. So if you cannot change forms, right? If hemoglobin cannot go from T to R, if hemoglobin does not have two forms, right? Then there is no cooperativity. So for cooperativity, you need two things. You need at least two conditions. Oops. Condition one, multiple binding sites. Binding sites. So to have cooperativity, my protein needs to have multiple sites where it can bind that ligand. And binding sites need to communicate. And I'm going to put that in quotes, right? Because as we saw, they don't really talk. There's just amino acid interactions that they can have, that they either form or they break, right? So these binding sites need to be able to communicate to each other. If one binding site binds a ligand, it can tell the other ones, hey, I'm bound to something. And remember, all that communication is the way it's done in hemoglobin. When oxygen binds, the iron is pulled towards the oxygen. When iron is pulled down, histidine gets pulled down with it. When histidine gets pulled down, the whole what's called the F helix is part of hemoglobin, gets pulled down. This causes a whole change in the tertiary structure of one subunit. This affects that one subunit, how it interacts with the other subunits, which has an overall quaternary changes. It changes the other subunits. So that was a kind of refresher on how hemoglobin works itself. But any questions about those ideas, how hemoglobin and the T and R states work and how that can affect our various properties? Well, if you're thinking of a question, how about D as well? How is BPG affected? I guess it should be affected. Anyways, if I'm in Rainier, does BPG have more of an effect, less of an effect, or does it stay the same? And I don't have a poll for that. That was just a question I thought of right now that I should have put on here to begin with, but I didn't. So for Rainier, will BPG have more, less, or the same? So I have a more, I have a less. So remember what BPG does. BPG binds to the center of hemoglobin and stabilizes the T state, right? So it stabilizes the T state. However, if I have a mutant hemoglobin that stays in the R state, BPG, there is no T state for BPG to bind to, so it's less of an effect. Let me just go back. So if we just go back a slide, 2 slides, here, when BPG is binding right in the center there, that type of binding is only possible in the T state. And I have here, when we go to the R state, BPG is actually kicked out of the center cavity because it doesn't bind to the R state. So if we're mutated and we stay in the R state, right, then then BPG won't ever bind. So it won't have any effect. So you can see if you have a mutant hemoglobin like we have in Rainier, that causes a lot of problems. So any questions about that? This is a very good review question of how does hemoglobin work. And just quickly as a reminder, in case you forgot what the answers were, A is oxygen affinity increases, Bohr effect is less effective, Hill coefficient decreases to 1. BPG decreases. It will stay, it won't bind to hemoglobin, right? So the effect of BPG decrease, the amount of BPG, that might change. But if our hemoglobin never goes to the T state, BPG has no effect on our hemoglobin. Five is another one of those fun thought questions. I'll do it really quick because I actually like this question. So the reason why when we're like drowning or holding our breath that we have that sudden urge to breathe, it's not because lack of O2. Your body has no way to determine how much O2 is in your body, right? There's no oxygen sensors, blood oxygen sensors. So we have no idea, your body has no idea how much oxygen's in your blood. What triggers us is the amount of carbon dioxide in our blood. So we do have sensors for the amount of carbon dioxide in our blood. And so when the carbon dioxide in our blood goes high, that's when we feel like, oh no, we need to breathe. Because if carbon dioxide's high, that means oxygen's probably low. Now, free divers are people who dive very deep in the ocean without oxygen tanks. Now, one belief of free divers is that they will hyperventilate just before they make a dive because they think that hyperventilation will increase the amount of oxygen in their blood. Does that actually happen? And I will just answer that because we did talk about it on Friday, and this is just one of those fun questions to understand how the body works. And by hyperventilating before you dive, you are actually decreasing the amount of oxygen in your blood, right? So if you breathe really fast, you're not intaking oxygen like that well. What you're doing is that you are basically causing hypoxia to your brain. You're decreasing the overall amount of oxygen in your blood. And like I said, we talked about this last week, but if any of you ever want to become a free diver, or any of you are a free diver, generally you don't want to hyperventilate before you go and hold your breath, because that will decrease the amount of oxygen you have in your blood. So that is just opposite. Basically, by breathing that fast, you remove carbon dioxide, and then, yeah, it doesn't quite work that way. Yeah, you increase the amount of carbon dioxide in your blood. But yeah, that's just connecting hemoglobin to like some fun, social science. Next time you're at a party when COVID's over, remember when parties were a thing, when people got together? Patrick Farms remembers. But if parties ever become a thing again, that's a fun little party anecdote. You know what I learned in biochemistry about free divers and hemoglobin? I mean, some interesting stuff. Anyways, question 6 is again connecting hemoglobin to like an actual physiology. So question 6, I think we're gonna like skip as well because it doesn't really talk about how hemoglobin works. It's just, getting at the idea of how biology and chemistry can be connected. So again, I'll just go over it because I think it's a cool idea, but I'm not going to ask you a question like this on the test, where we have an individual whose blood only produces half the amount of hemoglobin. And just by looking at them, they look in good health. However, so they're all in good health. However, if you take a normal individual and you give them enough carbon monoxide to bind half their hemoglobin, they die. Why is that? So I'll just write that out. Like anemic individual, let's say a normal individual has like 100 hemoglobins and a normal individual only has 50 hemoglobins. They are fine. But if I add CO, so now half my binding sites are bound, this individual dies. And what that question's really getting at, it's really an odd question. It's one of those thinker questions, is that hemoglobin has 4 binding sites, right? Binding site, binding site, binding site, binding site. And so it says occupy half of her heme sites, right? So if I have 100 hemoglobins and half are bound, so carbon monoxide, carbon monoxide, carbon monoxide, that could mean all 100 hemoglobins are affected, right? Because each hemoglobin has 4 sites. And if one carbon monoxide binds there, that could wipe out the entire hemoglobin. The entire hemoglobin would have a hard time delivering oxygen now. So really, this question is really getting you to understand what does it mean for a binding site for hemoglobin. And the main thing I want to get out of that is, remember, hemoglobin has 4 binding sites. These binding sites need to talk to each other to transition from the T to R state. And that is how we effectively deliver oxygen. That's the main takeaway from this problem. And unfortunately, due to time, usually what would happen is that we would sit and talk about this for 5 to 10 minutes. But this is one of the places where I can make up time for this class. So that's why I'm going to do it. But yeah, that's the main takeaways. And that is actually the end of this PowerPoint right here. So hemoglobin, all we talk about hemoglobin here is what we're going to need for the test on Friday. So basically, carbon monoxide will never leave the site, right? And so when carbon monoxide binds, it transitions hemoglobin like to the R state, because basically the hemoglobin really doesn't know if that iron molecule gets pulled down, like to interact with carbon monoxide, it will transition from the T to the R state. However, that carbon monoxide binds so well, right? It will never want to leave. It never wants to leave that binding site. And so we're never going to get that transition to T, right? And so that binding site will forever be bound. And then that hemoglobin will like forever be in the R state, which means that it doesn't want to let go of oxygen. So that's just one reason why that's super bad for you. And so not only is the amount of binding sites decreased, but now your heme is not going to transition to the T state. And that T state transition is where we let go of oxygen. T state's good. We need the transition between T and R because that's high affinity and low affinity. That's the answer in a nutshell. But like I said, I'm not going to ask you like that question, but I do want you to know like that main information, right? And I guess these questions are good examples of the type of thought questions I might ask you on the test. Like, yeah, I might give you a thinker like this. I won't give you many thinkers. I like to give you a couple. All right, let me transition to my next PowerPoint. We are done talking about heme. No more heme. We are now going to talk about muscles. Yay. Okay. So before we talk about how muscles work on the biochemical level, like the protein level, which is what we're ultimately interested in, we have to know how proteins work on like the big zoomed out A and P level. So I just want to say for now, I've never taken A and P in my life, right? Because I was a chemist. So those of you who have taken A and P, you probably know this stuff better than me. I know the biochemical stuff of this so well, but when we get to the big picture stuff like this, it's like, I'm more interested in proteins. But we do have to cover the big picture stuff. So muscles are made out of long multinucleated cells. They're called myofibrils. These run about 20 to 100 micrometers. A micro is times 10 to the minus 6. So they're big, 20 to 100 micrometers in diameter. and one cell runs the entire length of your muscle. That's kind of incredible when you think about it. You have like this one cell that can stretch your muscle, right? When we look at these muscle cells, we'll see that they are striated. That is, they have different colors, and these colors will repeat. And so we call these things by their colors. So the light colored area is called an I band. So this is the I band. While the darker colored area is called the A band. So we have the A band and the I band. 1 unit of the muscle runs I band to I band. We call that unit a sarcomere, right? When we look at the sarcomere, the I bands are anchored to what's called the Z disc. So the Z disc anchors your I bands, right? The A band, the dark line in the middle of this A band, we call that the M disc. So M disc is in the center of the A band. And that little light colored area right here, we call that the H zone. So a lot of random just letters thrown out there. But we will break down what's in the I band, what's in the A band on a protein level here in a minute. So the A band, the dark area, we call that thick. So the dark area is the thick filament. The lighter area, we call that the thin filament. So we have 2 different filaments we care about: thick filament and thin filament. That's the main thing we are caring about here. We have thick filament and thin filament. I is in the word light, A is in the word dark. Hey, that's a pretty good memorization tool. I like it. Light, dark. Very good. Thank you for sharing. I never thought about that before. All right, so we have I band, A band. So here's just our first question as a thought experiment. So in our muscles that are striated like this, when cells undergo mitosis, they don't do cytokinesis. So they don't split, which gives you these huge cells that are multinucleated. That is, you have these very long cells with a ton of nuclei in them. Why do muscles do this? Or another way to put it, why is it, would your muscles probably be less effective if they did do cytokinesis? Does anyone know the answer to that, or want to propose an idea that would answer that for us? Here's a good critical thinking question. Something that's not obvious. There would be too much going on with each individual cell. Please be a little more scientific in what you mean by too much going on. That is too vague. Too much room for error. Can't control from a distance either. There is room for error. Yep. So let me try to get at the heart of this, right? So these cells, right? If we look on this image, let me get my pen out with a different color. Red will do just fine. If we look at this image, these cells, and I cannot draw it as well as these cells are. These cells are nice and they're lined up, right? And you have these nice lines stacked on each other. And that's how the muscle works. You have a bunch of different lines, right? If we look at the individual cells here, right? They're all lined up. Now, imagine how much more difficult it would be to form these nice lines. Instead of these long spaghetti cells, you had individual cells. And you tried to connect them all through extracellular proteins. That does leave a lot more room for error, right? And so the reason why, we have these long cells really gets to the purpose of the muscle, or rather, why the muscle cells look the way they are, that they're positioned end to end in this linear format. So in shorter cells, it's going to be much harder to line up your cells end to end. That's basically what's going on. It's for ease of lining up your cells end to end to form these long muscle cells. Short cells, yes, more error. Hard to have them line up perfectly like we see in our image here. All right. So moving on. Here's the stuff we as in me actually care about. Well, as this class, this is what we're going to focus on more. It's the proteins that make up your muscles. So first we start with the thick filament. Thick filament is made out of myosin, right? So myosin is thick. Now let's look at the protein structure of myosin. It's made out of 6 peptide chains, right? So we have 6 peptide chains forming one protein. We have 2 heavy chains. Let me get my pen out. We have 2 heavy chains. So we have the blue, and this is also the other color there is also a heavy chain, right? So the heavy chains are wrapping around each other to form like a coil. So the heavy chains are wrapping around each other to form a coil. And then you have 2 heads, right? So each myosin has 2 head groups, and those are from the heavy chain. And the end terminus of the heavy chain is the head. The C terminus is the tail. Around the heavy chains, you have 4 light chains, and these come in pairs. So you have 2 pairs of light chains, or 4 light chains, right? And these bind around the end terminus of the myosin. The C terminus is a pseudo repeat, like we talked about for our other fibrous proteins. So A, B, C, D, E, F, G are just proteins. A and D would be nonpolar. And remember, it's the nonpolar areas that are making these proteins stick together, right? Because the hydrophobic effect pushes nonpolar amino acids together. So to have this winding, have this interaction, you want these nonpolar amino acids to come together. When we look at myosin in the structure of muscle, what we see is that they're kind of lined up, right? So each one of these pair of heads, that's myosin. So you have an area that's full of myosin heads, but then you have a bare zone, right? This is where there's nothing but tails. So when we look at myosin inside the muscle, you'll find this configuration where you have a bare zone of myosin tails, and then you have areas of myosin heads. So all of these myosins come together to form the thick filament. So any questions about myosin? So our thick filament is made out of the protein called myosin. All right. Question? So question is, that's the whole muscle. what we're seeing on that image at the bottom is the whole thick filament, right? So let me get rid of my drawing. So like myosin, right, makes up the thick filament, right? So it's like making up this A band where we have nothing but myosin heads. So the bare zone is kind of like the H zones. And then like we have here, oh, I'm using my mouse. And then here we have like the myosin heads, right? But this are all those myosins more or less connected. That's kind of what's going on there. So these are your myosin. And so have this dark image in your mind, and then we go back, and it's actually, that's what it is. It's proteins. So what happens when we tear a muscle? What happens to the tail? Something say don't do my. So these are just proteins, right? These aren't cells that we're looking at. This is just protein inside of a cell. And I think when you tear a muscle, you just need to repair those cells. I'm not exactly sure what happens because, like I said, I study I know about the proteins inside. Tearing the muscle, that's more of a A&P type of question. So I actually don't have that information on the top of my head. So A and D are what hold the second filament together. A and D are what's holding these tails together, right? So you have hydrophobic and hydrophobic. They all match up with like hydrophobic and hydrophobic amino acids. So when these things are interacting, they're like interacting at A and D more or less to try and help them come together. Yeah, so A and D are just amino acids in the tails, right? And so the tails are wrapping around each other. And to facilitate that wrapping, A and D are hydrophobic amino acids. Because of the hydrophobic effect, water will try and make hydrophobic amino acids come together. And so that's why they can wrap around each other. So that A, B, C, D, and E, they're A, B, C, D, E, F, G are just amino acids. So remember, we are looking at protein. So we've gone closer than the cellular level right now. So we've gone into the cell of these long muscle cells to see what's happening on a protein level to make our muscles work. That's what we're looking at, right? So we started big here where we're looking at the muscle cells. We see that they're colored differently. So what's going on? Okay, let's look at the proteins that are making these muscle cells colored differently now. So that's where we're at. So just as a little refresher, because we talked about this previously a couple weeks back, would you call myosin globular, or would you call it fibrous or fibrous? I do have a poll for this. Would you call it myosin globular or fibrous? Fibrous, fibrous, potato, potato. Does anyone actually say potato? I have to think that's made up for the song. I have never heard anyone seriously go like, yeah, I got myself a bag of potatoes. I think that's a made up word. All right. So we have 10 votes for fibrous, 3 votes for globular, and the correct answer is an option I didn't give you. Sorry, I was being mean. It's both. So here's we talked about like 2 weeks ago how we have 2 different classes of protein, where you have a fibrous protein and a globular protein. So fibrous is like a long, like alpha helixes. Globular is soluble alpha helixes and beta sheets. Myosin is made out of both, right? Here's fibrous, here's globular. So trick question. I would never ask you something like that on the test and not give you the correct answer. That's just me being unfair to you. So the correct answer is both. All right. So we learned about the thick filament, what the protein makes up the thick filament. Let's look at the protein that makes the thin filament. So remember, myosin is thick, actin is thin. And so the thin filament is made out of actin. And actin is actually the most abundant protein in your cytoplasm because actin is what makes up your cytoskeleton. So actin is in every single protein or every single cell, right? Every cell that you're going to look at is going to have actin in it. Here, for muscle cells, something else actin does is that it makes the thin filament. So I'm just going to go back a couple slides, right? So the myosin is the dark color, the actin is the thin color. Head is globular, tails are fibrous. Yep. So blue and green are globular, more or less. And the red is fibrous. Yep. Yeah. So actin has two different forms. We call it G-actin and F-actin. Now, this is a common confusion that I see. G-actin and F-actin are not different proteins. G-actin and F-actin are the same protein, just in different states. So you can think of G-actin and F-actin like T and R hemoglobin, right? So T and R hemoglobin is the same protein, different conformations. G-actin and F-actin is the same thing, right? So if actin is all by itself, that is, it's in its monomeric form, it's not bound to other actins, we call that G-actin. So if it's a monomer, not bound to other actins, it's a G-actin. But these G-actins can combine to form F-actin. So when I was a kid, Power Rangers were super popular. And so I don't know if anyone's ever seen Power Rangers, but what happens is that each like little soldier guy, he has his own robot. And then the robots come together to form the Megazord, which is a giant robot. That's basically what happens here. The G-actin acts independently and by itself, but then it can come and combine with other actins to make F-actin. So that's what's happening here. So it goes from globular to a fiber. Yes, Mighty Morphin Power Rangers for sure. I was a big fan when I was like 6. All right. So when we're looking at actin, actin has binding sites. Let me undo some of my drawings. So actin has binding sites for ATP, calcium, and magnesium. And what happens is that G-actin has ATP. And then when we form into a fiber, after we form that fiber, that ATP is going to hydrolyze, and we're going to make ADP. So G-actin will have ATP bound. F-actin generally has ADP bound. And what F-actin looks like DNA more or less. That's F-actin over there. And it forms a double helix. So you have this double helix of actin. And what's interesting is that there's polarity with actin. So you have a plus end and a minus end. So just like how DNA has polarity, actin has polarity. On the plus end, actin adds faster, right? So actin can add on to either the plus end or the minus end. it will add faster on the plus end, and it will come off faster from the minus end. So what actin looks like, it's like a moving treadmill where like, yeah, it's a treadmill that moves where like part of it will disappear from one end, but the other end will keep building. Oh, that's the wrong color. And so that's how it can move across the cell while being F-actin. It loses from one end and gains on the other end. And F-actin is the one that's binding the myosin. So if we go back to our muscles, our image of the muscle, right, where we have the thick and thin filaments interacting, the thick filaments, which are myosin, right, the myosin heads are binding the thin filaments, which are actin. Super Robot Monkey Team Hyper First Go. That is, I was already too old for that because I have no idea what that is. The last cartoon I remember being popular among people my age were Powerpuff Girls. And even then I thought I was too old for that. And that was like 7th grade. But any questions about F-actin or G-actin or any of the information that is presented on this slide, any of the topics I just went over there? F-actin is thin filament, correct. 13-year-old. What does it mean in context? Oh yeah, I kind of skipped over that, didn't I? When you're in F-actin, right, one actin is touching like 4, because it's a double helix. So one actin in the rope is touching 4 other actins at a time. That's what it means. And that's there. All the magic happens with power force. All the magic happens because of ATP, I guess. If you're talking about the power stroke, we'll talk about that in a minute. How does the pattern repeat if only one side binds to myosin? So F-actin binds to myosin, right? And it's not that the plus N or the minus N binds to actin. It's that when actin's in the fibrous form, myosin binds to that, right? Myosin doesn't really bind to G-actin. I don't know if that answered your question. And generally, with the thin filaments, you don't have treadmilling all that much. Treadmilling that I was talking about happens with like actin in the cytoskeleton. Like that's how, if you ever seen those E. coli move like on a slide, that's because of its cytoskeleton rearranging and all that good stuff. That's actin. That's what actin's doing there. It's not referring to charge. It's referring to how fast we're adding actin onto the side. The positive end, you get actin adding fast. The negative end, actin removes fast, right? So actin itself, like, it doesn't mean that one end is positively charged and one end is negatively charged like a protein. All that means is the speed at which actin is adding or actin subtracting. No, so I thought G-actin localized on one side and F-actin on the other. No, so it's just, am I in a fiber? Then I'm F-actin. Am I all alone? Then I'm G-actin. The thin filament is going to be F-actin. Okay, question 3 is more of review, so we'll can just pick that up on Wednesday, and then we'll get on to the power stroke and talk about rigor mortis. So as always, I will put up a new homework. I'll put this video up. This stuff, though, just as a repeat from beginning of class, all of this is not on the test. So this whole skeletal stuff, not on the test. This is on test 3. This is test 3 material. So test 2 will end at hemoglobin. So if you have any questions about, any of the material in test 2, please shoot me an email, all that good stuff. Are we going to have any math solving problems? Yeah, right. Hemoglobin. We did a bunch of math with hemoglobin. That's what I'm thinking off the top of my head. I have the equations provided. I don't know. I haven't made the test yet. Let me make the test first, then I'll get back to you. Yeah, so that's TBD, to be decided. Otherwise, if you have any questions about the test material, let me know. Yeah, as always, have a good rest of your day. I will see people on what? Wednesday. Wow, I can't believe it's the end of Monday already. Take care.